Vitamins and food. The course outline for this module starts with the classification of vitamin based on the sources and structures, bioavailability of vitamins, effect of different types of processing on vitamin and retention strategy of vitamin food. Therefore, at the end of the lecture, the students are expected to be able to classify different types of vitamins, explain the chemistry and functions of different types of vitamins, and comprehend the effect of food processing on stability of vitamins. Vitamins are minor components of food and present in natural food. Vitamin is a nutrient that we require in very small amounts that we cannot produce ourselves, but we need it for normal growth, maintenance, and functioning of metabolism. When absent from diet, deficiency disease can occur as it cannot be synthesized by human. Moving on to the classification and sources of vitamins. Vitamins can be classified into fat-soluble and water-soluble vitamins based on their solubility property. Fat-soluble vitamins are vitamin A, D, E, and K, while water-soluble vitamins are vitamin B and C. This list shows the classes of vitamins with their active forms. Fat-soluble vitamins are absorbed through the intestinal tract with the help of lipids and can be stored in our body because our body is surrounded by fatty layers that allow the movement of those vitamins and storage. Vit is mainly stored in the liver. Vit K and E are partially stored in the liver. Vit D is mainly stored in the fat and muscle tissue. Since they are more likely to accumulate in the body, they are more likely to lead to hypervitaminosis than are water-soluble vitamins. Meanwhile, water-soluble vitamins dissolve easily in water and, in general, are readily excreted from the body. Excesses are excreted in the urine since they are not readily stored. Consistent daily intake is important. This diagram shows the digestion and absorption of vitamins. In A, vitamins are initially released in the stomach upon eating of food containing the vitamins. In the small intestine, the fat-soluble vitamins are transported into the intestinal cells as part of micelles. Once inside the intestinal cells, fat-soluble vitamins are packaged with fat and other lipids into chylomicrons that travel through the lymph system to the main circulation. For water-soluble vitamins, they are absorbed directly into the portal vein from the small intestine. Table 4 summarizes the important vitamins with daily requirement. Chemical name or active form, biological functions, deficiency and excess symptoms and sources. For dietary recommendation, please refer to the Malaysia Guidelines, Recommended Nutrient Intake 2017. For vitamin A, beta-carotene is a precursor to vitamin A, which we will discuss later on the chemical forms of vitamin A. This is a continuation for Table 4. Vitamin B9 or folic acid is essential to be taken by pregnant women as the deficiency in folic acid can cause neural tube defect. Meanwhile, vitamin C plays major roles in maintaining our healthy body system. Vitamin D or ergo in cholecalciferol is derived from the conversion of sunlight on 7-dehydrocholesterol, which will be details out later. Vitamin A can exist in different forms, depending on the functional group. Retinol ester is storage form of vitamin in our body liver. It is interchangeable with retinol and retinol, which have alcohol and aldehyde groups, respectively. However, once the retinol changed into retinoic acid, this is an irreversible reaction that we want to avoid. As mentioned in the previous table, beta-carotene is a provitamin for vitamin A. What is provitamin? It is a compound which is not a vitamin but can be converted in the body to a vitamin. In this figure, 1 molecule of beta-carotene could yield 2 molecules of vitamin A. The enzyme 15-deoxygenase can cleave a beta-carotene symmetrically to procure 2 molecules of vitamin A. This enzyme occurs in our intestinal mucosa. Other provitamins are alpha and gamma-carotene and cryptic xanthan. The main way that we get vitamin D is through UV exposure or our exposure to sunlight, so this is going to be the first step in the synthesis of a precursor vitamin D to an active vitamin D. We can get some vitamin D from the food that we eat from animal and plant products, but it's quite minimal. The exposure to UV light is going to trigger the conversion of a cholesterol that's present in our skin and our epidermis called 7-dehydrocholesterol to vitamin D3 called cholecalciferol. Cholecalciferol will now be floating around in our bloodstream and ultimately going to get to our liver. In our liver, cholecalciferol comes across an enzyme called 25-hydroxylase, and what it does is it gives a hydrogen to the 25th carbon of cholecalciferol to turn it into 25-hydroxycholecalciferol, which is also known as calcifediol. If we ingest vitamin D from animal products, which is fish predominantly, this comes in the form of cholecalciferol or vitamin D3, and therefore it's just kind of enter the bloodstream and go via this pathway in the liver. Vitamin C is a highly polar compound, readily soluble in aqueous solution that mainly influence oxidation reduction reaction in all living tissue. And vitamin C in the form of ascorbic acid is readily reversibly oxidized to L-dehydroascorbic acid or DHAA through oxidation. Further hydrolysis reaction will turn DHAA into a reversible form of dictogulonic acid. Vitamin C has several important roles in food industry. It applies in food to inhibit non-enzymic and enzymic browning by chelating metal ions and reducing oxygen. It can make each compound unavailable for reacting with the phenols. Another function of vitamin C is in dose system, where ascorbic acid improves bread texture and loaf volume by oxidized into DHAA. The reduction of DHAA back to AA drives the reaction of sulfhydryl compounds and the gluten to form intermolecular disulfide bonds. It is the network of disulfide bonds formed in the gluten structure that enables the dough to retain carbon dioxide produced by yeast, which in turn allows for maximum volume and improved texture. Bioavailability is the degree to which an ingested nutrient undergoes intestinal absorption and metabolic function or utilization within body that may be considered the relative absorption of a nutrient from the diet. It is an index of bioavailability may be extended to include the relative accumulation of a nutrient into various tissues. Various nutrients and dietary components interfere with the bioavailability of vitamins. Hence, requirements for vitamins cannot be considered independently, but must be evaluated in relationship to other nutrients and compounds consumed by an individual. Complete description of nutritional adequacy of food must be measured based on several parameters, including concentration of the vitamin at the time of consumption, various chemical species of the vitamin present, and bioavailability of these forms of the vitamin as they exist in the meal consumed. Table 3 shows the bioavailability of vitamins and minerals from vegetables and fruits in human food processing, such as fermentation, can enhance the bioavailability of some vitamins such as vitamin K. Moving on to the factors that contribute to the variation or losses of vitamins in food. Plant physiology, such as genetic cultivar and stage of maturity, site of growth and climate, post-harvest and food processing. Table 8.5 shows the influence of degree of maturity on ascorbic acid content of tomatoes. The highest ascorbic acid content was reported in yellow-red tomatoes aged 5 weeks from anthesis. Pretreatments such as peeling and trimming of fruits and vegetables can cause major losses of vitamins, as they are concentrated in the discarded stem, skin, or peel fractions. Alkaline treatments to enhance peeling can cause increased losses of labile vitamins such as folate, ascorbic acid, and thiamine at the surface of the product. Losses of this kind tend to be small compared to the total vitamin content of the product. Milling of cereal grains involves grinding and fractionation to remove the bran and embryo. Since vitamins are concentrated in the embryo and bran, major losses of vitamins can occur during milling. This becomes the rationale for the initiating the enrichment of cereal grain products with several added nutrients and folic acid. In post-harvest, fruits, vegetables, and animal tissues often retain enzymatic activities that contribute to post-harvest changes in the vitamin content of foods. For example, oxidative and hydrolytic enzymes can cause deterioration of cellular integrity and enzymatic compartmentation and changes in the distribution of chemical forms and activity of vitamin. Post-harvest changes in vitamin concentration are inevitable but can be minimized with proper post-harvest handling of fruits and vegetables. Mishandling of plant products through prolonged holding or shipment at ambient temperatures can contribute to major losses of labile vitamins. Continued metabolism of plant tissues post-harvest can be responsible for changes in total concentration as well as distribution of chemical forms of certain vitamins depending on the storage conditions. Blanching, a mild heat treatment, is an essential step in the processing of fruits and vegetables. Primary purposes are to inactivate potentially deleterious enzymes, reduce microbial loads, and decrease interstitial gases prior to further processing. Inactivation of enzymes often has a beneficial effect on the stability of many vitamins during subsequent food storage. Blanching can be accomplished in hot water, flowing steam, hot air, or with microwave. Losses of vitamins occur primarily by oxidation and aqueous extraction, with heat being a factor. Figure 8.2 shows the retention of ascorbic acid in peas during experimental water blanching for 10 minutes at different temperatures. Leaching of ascorbate in peas into water can be clearly visualized based on the decrease in ascorbate in peas while increased percentage of ascorbate in water at 20 to 100 degrees Celsius. This figure shows the effect of storage at different temperature on the ascorbic acid, dehydroascorbic acid, and total amino acid content of fresh-cut kiwifruit slices. At higher temperature of storage condition, the amount of ascorbic acid is the lowest with the highest conversion to dehydroxyascorbic acid. The table shows the effect heating time on the concentrations of vitamin C in several vegetable samples. A longer heating time will cause higher loss of vitamin C in vegetables. Therefore, to maintain vitamins during processing and storage is surely a daunting task, as it involves the control of many variables, which includes duration and temperature of processing or storage, concentration dependence and temperature dependence of the degradation reaction, environmental variables such as pH, availability of oxygen, metal ions, and reducing and oxidizing agent, rate of competing or sequential reactions, relative stability of the various forms of the vitamin. chemical nature of other food components, e.g., reducing versus non-reducing sugars, mechanism of chemical loss of vitamin activity in the food, water activity of the food system. To conclude, vitamins are crucial micronutrient in human diet for various important functions. We have different types of vitamins include water and fat soluble. Various nutrients and dietary components interfere with the bioavailability of vitamins. Stability of different types of vitamins is affected by many factors and variables. These are the references. Thank you.